Today we will discuss about mutation and its classification. What is mutation? A mutation is an abrupt inheritable change in the genetic material of an organism that may be both qualitative and quantitative. It is the change in the DNA sequence that in turn changes the corresponding RNA and protein synthesis. It involves only one base or a base pair of DNA and can occur in random manner and is not directed according to the requirement of organism. Most mutations occur spontaneously by environmental effects but can also be induced in the laboratory using mutagens. Mutations occur frequently in nature and have been reported in many organisms like Drosophila, mice, and other rodents, and man. In the Drosophila, mutation causes white and pink eyes, black and yellow body color, and vestigial wings. In man, the mutation causes variation in hair color, eye color, skin pigmentation, and several somatic malfunctions. Various genetic diseases of human beings, such as hemophilia, color blindness, phenylketonuria, etc., form other examples of mutation in human being. So, any change in the sequence of nucleotides in DNA result in the corresponding change in their mRNA and in turn their protein synthesis due to the transcription and translation of the nucleotides. These defective enzymes or structural proteins may adversely affect the trait controlled by the protein, and mutant phenotype make its expression. And a classification of mutations. Classification is based on different aspects. First, according to the occurrence in the somatic and germ cell line, they are classified into somatic mutation and gametic mutation. Somatic mutation is the one that is occurring in the non-reproductive body cells. That is, it occurs in any body cells except for the reproductive cells. And usually they are related with malignant growth, that is the cancerous growth. A gametic mutation is the one that occurs in the gamete cells, that is the sperm and ova, the reproductive body cells, and these are heritable and are of immense genetic significance. Next, they are classified based on the size and quality. That is point mutation. In point mutation, there is an alteration in the small segment of DNA molecule, that is a single nucleotide or a nucleotide pair is altered. It is further classified into substitution, insertion or addition and deletion. In substitution mutation, the nucleotide of the triplet is replaced by another nucleotide is called substitution. That is, it affects only a particular triplet codon, and the altered code word may designate a different amino acid and result in the production of protein with single amino acid substitution. This substitution reaction is further divided into two types, one transition and other transversion. Transition is when a purine base is substituted with another purine, that is when adenine is substituted with guanine, or it is when a pyrimidine is substituted with another pyrimidine, that is when a thymine is substituted with cytosine. These kind of reactions are called transitions, and it occurs due to tautomerism. So, when normally a purine like adenine and a pyrimidine thymine is usually bound by two hydrogen bonds, while the guanine is linked to cytosine with three hydrogen bonds. This is a molecular configuration that is common in every base pairing, but in some uncommon molecular configuration, the single proton shifts is generated and are called rare states or tautomers. A tautomeric shift is believed to occur when an amino, that is an NH2 form of adenine, is changed to imino, NH form. This is the tautomeric shift and may occur in thymine, changing it from the keto, that is C double bond O form, to enol, COH form. The another reason for transition may be the abnormal pairing due to transitional substitution due to ionization of the base at the time of DNA replication. This ionization is the loss of hydrogen from the number 1 nitrogen of the base. That is, it is in its ionized state, thymine pairs with guanine if guanine is in its common form. In a similar fashion, guanine in its ionized state can pair with thymine in its common form. The next type of substitution mutation is transversion. That is, a pyrimidine is altered with a purine, or a purine is altered with pyrimidine. These type of reactions are called transversion reactions. Deletion reactions, where there is a loss of some portion or in a single triplet codon, is called a deletion mutation. It is frequently reported in phage T4, that is bacteriophages. Insertion mutation, it occurs due to addition of one or more extra nucleotide to the gene. The mutation which arises from insertion or deletion of individual nucleotide and cause the rest of the message downstream of the mutation to be read out of phase is called Frameshift mutation. That is, if there is a change in one nucleotide, the rest that is read downstream of the mutation is read out of phase, and this is called frameshift mutation. And the result is the production of incorrect inactive protein that may also result in death of the cell. Next, the mutation is classified according to change in amino acid sequence. One is missense, wherein there is a change in the meaning of the codon by changing one amino acid into another. That is like shown in the picture. When A is changed to U, it forms a leucine. Temperature sensitive mutation or TS mutation is the substitution produces a protein that is active at one temperature but becomes inactive at a higher temperature. Nonsense or chain termination mutation. This is occurring when there is a mutation to a termination codon, resulting in production of shorter protein. That is, when the codon is changed to UAG, UAE or UGA, which are stop codons of the termination codons, it is termed as nonsense or chain termination mutation. Silent mutation. This changes the nucleotide, but the amino acid sequence are not affected. Because of the Obel hypothesis, it is understood that the third codon is not much important. Hence, the change in this third position of the codon doesn't alter anything, and this is referred to as silent mutation. Then we have classification based on the type of chromosome present, that is autosomal mutation and sex chromosomal mutation. Autosomal mutation occurs in autosomal chromosomes, whereas sex chromosomal mutation occurs in sex chromosomes. So if you like the video, please do share, like and subscribe to our channel. Thank you. For detailed notes, you can click on the given link below.